Hello, my name is Joshua Gavi. I welcome you to Coordination and Control. Question 26 to 30. Why is cyclic AMP referred to as a second messenger? Part B, compare tropism and nastic response. Roman number 2, plant and animal hormones. Explain why cyclic AMP is referred to as a second messenger. CAMP acts as an intracellular chemical messenger, activating specific enzyme, catalyzed reactions, cascade reactions, after it has been produced in the response to hormone, first messenger, that attach on the receptor site of the cell surface membrane. Part B, compare tropism and nastic response. Let's start with the similarities. Both are plant responses to external stimuli. 2, both are coordinated responses involving either hormones or electrochemical messengers. Both may involve growth curvatures. Let's look at the differences. Nastic response depends on external stimuli, and tropic response depends on the election of the stimuli. Nastic response involves growth or tag changes. Tropic response involves growth only. Nastic response are relatively fast. Tropic response are relatively slow. Nastic response depends on electrochemical messengers. Tropic response depends on hormones like auxins. Nastic response takes place in any part of the plant. Tropic response occurs in growing parts of the plant. Part B, compare plant and animal hormones. Let's start with the similarities. Both are organic substances. Both act in low concentration. They exert their effect on other parts of the organism other than where they are made. Differences. Plant hormone mainly exert growth effects. Animal hormones, these affect a number of other physiological effects other than growth. Plant hormone, they are produced from unspecialized cells. Animal hormones, these are produced from specialized cells of the endocrine tissue. Plant hormones exert effects quite close to their site of production. Animal hormones, they exert effects at a distant site from the site of production. Plant hormone lacks specificity, influence different tissues and organs, sometimes in contrasting ways. Animal hormones affect specific target areas and have no effect on other organs. Plant hormones, they tend to interact with each other and in control of growth. Animal hormones often act singly and bringing about a specific change. Number 27. Outline the effect of light via phytochrome system on the different physiological processes in plants. Part B. Compare the effects of the various phytohormones. Part A. Outline the effect of light via phytochrome system on the different physiological processes in plants. Let's look at germination in small seeds. Effect of far red light and of darkness inhibits germination of small seeds. And effects of red light and of white light. It promotes germination of small seeds. Let's look at the growth of the internodes. Effect of far red light and of darkness. It promotes growth of internodes. An effect of red light and of white light. It inhibits growth of the internode. Development of chlorophyll. Far red light and of darkness inhibits development of chlorophyll. Red light of white light promotes the development of chlorophyll. Chlorophyll synthesis. Effect of far red light of darkness inhibits chlorophyll synthesis. Effects of red light of white light promotes chlorophyll synthesis. Leaf expansion. Effect of far red light of darkness inhibits leaf expansion. Effects of red light of white light promotes leaf expansion. Lateral root growth. Effect of far red light and of darkness promotes lateral root growth. Effects of red light of white light inhibits leaf root growth. Flowering in long day plants. Effect of far red light of darkness, it inhibits flowering in long day plants. Effects of red light and of white light promotes flowering in long day plants. Flowering in short day plants. Effects of far red light and of darkness promotes flowering in short day plants. Effects of red light, of white light, inhibit flowering in short day plants. Leaf fall, deciduous species. Effect of far red light, of darkness, promotes leaf fall, deciduous species. Effects of red light and of white light inhibits leaf fall. Part B. Compare the effects of the various phytohormones. Both auxins and gibberellins, they promote cell division in apical meristem. They induce parthenocarpy. They stimulate fruit growth, bring about cell enlargement, in return growth, stimulate flowering in long day plants. Cytokinin promotes cell division in presence of auxins. Both gibberellin and cytokinins promote lateral bud growth. Auxins promote apical dominancy, while gibberellins stimulate auxiliary root growth. Auxin promote flowering in short-day plants, while gibberellin inhibits flowering in short-day plants. Auxins promote phemeronase in flowers, while gibberellins promote melanase. Auxins promote root initiation from stem or leaf cuttings. Gibberellin inhibit development of adventitious roots. Phytochrome red inhibits stem elongation, while phytochrome far red stimulates flowering in long-day plants but inhibits it in short-day plants. Phytochrome red stimulates flowering in short-day plants but inhibits it in long-day plants. Ethylene and ethene, they inhibit cell elongation, while gibberellin promotes cell elongation. Abscisic acid stimulates bud dormancy, while gibberellin breaks bud dormancy. Lateral root growth is inhibited by phytochrome red and stimulated by phytochrome far red. Number 28. Explain what is meant by all or nothing law. 2. Refractory period. 3. Facilitation in relation to temporal summation. Part B. Explain the advantages and disadvantages of synaptic transmission. Explain what is meant by all or nothing law. The law states that the magnitude of the action potential generated is always constant, regardless of the amount of the stimulus applied, provided the stimulus is above threshold. Or if the strength of the stimulus is below a certain threshold intensity, no action potential is evoked. If, however, the stimulus is above the threshold, a full-sized potential is evoked and remains the same however much intensity the stimulus becomes. Number 2, refractory period. It is a period of inexcitability that accompanies the recovery phase of the axon. It lasts approximately 0.3 seconds. It is categorized into absolute refractory period, where the axon is completely incapable of transmitting an impulse, regardless the magnitude and the duration of the stimulus. relative refractory period, where it is possible to generate an impulse provided that the stimulus is no longer than normal, or is longer than normal. Refractory period is important because it determines the maximum frequency at which an axon can transmit impulses, ensures separation of action potential, and specify stimulus causing the excitation, as well as preventing spreading of action potential, and makes it flow in one direction. Part A. Facilitation in relation to temporal summation. Facilitation process involves the first impulse transmission being insufficient to trigger off an impulse in the postsynaptic membrane, but cause an effect. Therefore, the second faster stimulation will add to the former and generate an impulse at the postsynaptic membrane. The rapid repeated release of transmitter substance from several synaptic vesicles by the synaptic knob as a result of an intense stimulus produces individual excitatory postsynaptic potential, EPSP, which are also close together, that they summate and give rise to an action potential in the postsynaptic membrane. Part B. Explain the advantages and disadvantages of synaptic transmission. Advantages of synaptic transmission. Transmission or transmit information within the neuron. Direct impulse flow into one direction only. Sites for summation, both temporal and spatial. Site for integration of different sensory and motor information. Allow adaptation to a continuous and repetitive stimulus, which may otherwise cause damage to effectors. They enable the body respond to weak stimuli as a result of facilitation. They are physical basis of learning and memory. Disadvantages of synaptic transmission. Slow down the speed of transmission. Are highly prone to drugs and fatigue, which may inhibit impulse transmission. outline the core principles behind the human eye operates. B. Briefly describe the mechanism of accommodation in the human eye. C. Describe the structure and arthropods compound eye. Part D, Roman number 2, Part C. Roman number 2, reasons why having two eye binocular is better than having one. Part B, briefly describe the mechanism of accommodation of the human eye. Accommodation for the nearby object. We have an object here, one, and it produces diverging light rays which reaches the eye. Then the cornea here refracts the light rays, and then we have the ciliary body, ciliary muscles, they will contract. These ones will cause the suspensory ligament to slacken. And when the suspensory ligament slacken, now the lens will become more convex. When the lens becomes more convex, the light rays are focused onto the retina. When looking at a nearby object, the ciliary muscles in the ciliary body contract. The suspensory ligament slackens, and this makes the lens short and thick. This increases the ability of the lens to refract light and reduces the focal length of the lens for the nearby object to be clearly seen. So what is going to happen is that the ciliary muscles in the ciliary body will contract, and when they contract, the suspensory ligaments slacken. When they slacken, they make the lens to become short and thick. It becomes convex. And this one increases the ability of the lens to reflect light, bend the light more, and it reduces the focal length of the lens, thereby focusing the nearby object image onto retina. Accommodation of a distant object. When viewing a distant object, the ciliary muscles in the ciliary body will relax. This causes tension in the suspensory ligaments. The suspensory ligaments pull the lens apart, making the lens thin and long. This makes the lens to refract less and increases the focal length of the lens. So when the light rays are coming from a very far or distant ray object, they reach the eye in parallel. So the parallel light rays reach the eye. These light rays which we have, they will reach the eye parallel. Now when they reach the eye parallel, the cornea here is going to refract them, bend them a little bit towards the lens. Now the circular muscles are going to relax. When they relax, the suspensory ligaments become taut. And when they become taut, they are going to pull the lens. When they pull the lens is going to become thin. And when it becomes thin, it is going to refract the light, bend the light less. And when it bends the light less, it increases the focal length. And when it increases the focal length, it is going to be focused on the retina. Part C. Describe the structure of an arthropod compound eye. Now we have the compound eye here. These are called ommatidia. Each one is an ommatidia. And then here we have the fibers of the ommatidia with optic ganglions. And here we have the optic ganglion. This is the optic ganglion connecting to the optic nerve. Arthropod eyes or eye is a globular ovoid crystalline organ made up of numerous function islets called ommatidia. These structures that we are seeing, those are the ones we are calling ommatidia. Each structure that you see there is an ommatidia. Each ommatidium has elongated retinal cells surrounding the central placed light-sensitive rhabdom. The cone-shaped lens is capped by a flattened cornea, while the lower part borders the retinal cell, and the inside are surrounded by the lens-secreting cell. So when you look at the ommatidia here, this is the cornea. The whole of this one is ommatidia. And now what we have here in the center, this is the lens. And on the side here we have the lens producing cells. From the lens in the middle we have the pigment rhabdom, the light sensitive pigment. And then outside it, we have the retinal cells. The retinal cells outside it. And then the pigment cells are the ones outside. So when you cut across here, this is what you're going to see. Inside the center, you have the rhabdon, and then surrounded by retinal cells, and then outside you have the pigment cells. Then down we have connection of the fibers to the optic ganglion. These are the ones connecting to the optic ganglions. The rhabdon and retinal cells are connected to the optic nerve cells. As we have seen, this is the rhabdon in the middle, and then outside we have the retinal cells. These are retinal cells outside. All of them, they are connected to the, what we are saying, they are connected to the optic nerves. These are the optic nerves down. The rhabdon is formed of the concentration of microvilli from retinal cells. The ommatidia are separated from each other by varying degrees of pigmentation. So each one that you are seeing from here going down, that is an ommatidium. 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 So they are the ones we are saying they are separated by varying degree of pigmentation. Photoactive reactions in the rhabdon causes depolarization of the retinal cells. Part C. Outline reasons why having two eyes, binocular vision, is better than having one. Binocular vision gives a larger field of view. A defect of one eye does not result in blindness. That is, can be partly compensated by the other eye. It provides potential for stereoscopic vision for animals with two forward-facing eyes. Number 30. With 3 examples, explain what is meant by lateral inhibition and give examples of its effect on these 3 sensory systems. Part B, briefly describe the refraction errors of a human eye and how they can be corrected. With 3 examples, explain what is meant by lateral inhibition and give examples of its effect on these 3 sensory systems. Lateral inhibition is a central nervous system phenomenon by which the sensory information is sharpened by inhibition of the incoming neighboring or lateral field information from the peripheral of that region which is the maximally stimulated. The three sensory systems utilizing the lateral inhibition are senses of touch, cutaneous, hearing, and vision. In the skin, a single touch is felt when a blunt object touches the skin, because the surrounding fields have been laterally inhibited at the level of the central nervous system. In the discrimination of the different pitches of sounds with similar frequencies, neuroactivity is laterally inhibited so that the hair cells along the basilar membrane, which are maximally displaced by the sound waves, are selected for interpretation by the auditory cortex of the brain, while hair cells with neuroactivities from the surrounding region are suppressed. Lateral inhibition is also at work in the process of visual information at the level of the ganglion cells in the retina. Here, the receptive fields of each ganglion cell resembles a bull's eye, with the central core area and an outer surrounding area that oppose one another, that is, are antagonistic. Those ganglion cells that have on center fields are excited by light at the center of their visual field, while the surround is suppressed or inhibited laterally. Those ganglion cells that have the center field are inhibited by light in the center and stimulated by light in the surrounding due to distribution of these two types of ganglion cell fields along the retinal surface. Incoming light excites some and inhibits some of these fields, which translates later at the occipital cortex as sharper visual acuity. Part B. Briefly describe the refraction errors of human eye and how they are corrected. We shall start with short-sightedness, myopia. Short-sightedness or myopia, this is usually caused by a large eyeball or a very strong lens. Light from a distant object is focused in front of the retina. The individual can only see nearby objects, but not distant ones. This can be corrected by putting on a diverging, or what we call a concave lens. So So when light rays are coming from very far away, they can be diverged. And when they are diverged, then now they can be focused on the retina by use of a diverging lens. If you don't use a diverging lens, they are going to be focused in front of the retina. So that's why we use a diverging lens, because when they come parallel, so when we reach here, we don't want them to be focused in front of the retina. We put a diverging lens. This one will diverge them. When they are diverged, now they will be focused on the retina. Let's look at long-sightedness, hypermetropia. Long-sightedness, the hypermetropia, this is caused by a small or short eyeball or a very weak lens, so that a close object is focused behind the retina. Individuals can see distant objects, but they cannot see nearby objects. Long-sightedness can be corrected by wearing a converging lens, convex lens. So when light rays are coming from nearby objects, they are going to be refracted, focused behind the retina. So what we are going to do, we are going to put a converging lens so that they can be converged and then they can be focused on the retina. So if a person is long-sightedness, meaning he can only see things from very far, distant objects, but cannot see nearby objects, we correct that condition by wearing a converging or convex lens. Astigmatism. In astigmatism, this is caused by unequal refraction of the cornea and lens due to uneven curving in them. It results in some parts of the object being well focused on the retina and some not to be focused. It is normally due to old age. This can be solved by wearing cylindrical lenses. Presbyopia. Under presbyopia, this occurs when the lens hardens due to old age and does not focus, and can be corrected by wearing spectacles with convex lenses or often 2 pairs of spots may be necessary, i.e. a pair with convex lens for close vision and a pair of concave lens for distant vision. The 2 types of lenses can be combined into 1 pair of known as bifocal spectacles. Cataract. Cataract, it is a condition which occurs when the individual's aging. It caused by the eye lens becoming opaque due to thin covering formed on it. It is corrected by surgical removal of the thin opaque layer of the lens. Thank you very much for participating in coordination and control questions 26 to 30. Please do like, share, and subscribe.